So as Judy said, my appointment at Stanford is in neurosurgery, but I don't work on anything really related to neurosurgical interventions. What I do work on is neural stem cell biology and neurogenesis, and neurodevelopmental biology now is a major effort in my lab. We're very interested in how the immune system in the entire body interacts with and influences the brain, both at the level of adult behavior, but also in influencing brain development and functional outcomes in offspring. And my lab works on mouse children instead of human children, but it's really delightful for me to be here at the Mind Institute because so many of you work on the human counterpart to what I work on in the lab. And so I think there's a lot of interesting overlap between mice and humans. And I'm going to tell you a story about using a mouse model to try to understand a little bit more about how immune system, mother's immune system during pregnancy, might interact with or synergize with genes that confer risk of neurodevelopmental disorder. And today I'm going to talk to you about one of our candidate genes that's pretty well validated in autism and how it is influencing outcome when the mouse mom has a very mild immune event during early pregnancy. So without further ado, I'll get started with just a little bit of background about how do we actually become confident in what we know. And so this is a satellite view, sort of philosophical approach to some hardcore biology. So a lot of what we know is from what our parents tell us. No science behind it. It's just what grandma said, what mom said. Some things you don't really need science because we just know them. Like you don't need a study to know that pregnancy is related to having new babies, right? But there are other things that is less obvious, but learned through generations. How many of you have heard this one? Just 2 or 3? Well, almost all the women who might have had pregnancy. So this is actually a real thing. And in fact, my mother told me, because my wife had gestational colic, and said, wow, your baby's going to be really hairy when it comes out. And I said, what? And it was true. And she had a big mop of bright red hair. Turns out that, in fact, it is true. And if you look at the incidence of heartburn, severity of heartburn, and whether the baby has a lot of hair at birth or not, there's a very strong correlation. So here's folk wisdom that our grandparents, our parents are telling us that if somebody turns around and asks, well, is that really true? Yeah, it turns out to be true. So science validates something we already know. I think in science there are things that we accept but we don't really understand. So species similarity, we share a lot of genes, we have the same shape, and yet we are so different. I think there's a wonderful diversity within a species. Our children, the way they come out, you can imagine that if we didn't have this diversity, we'd be a pretty boring lot altogether. So I think this is really important. Physical diversity, there's other kinds of diversity that we could talk about. In children, there's cognitive differences, and sometimes you're gifted, the child is gifted. Most of us are normal, that's the definition of normal because most of us are that way. And then there are some that are challenged. And there are a lot of reasons why a child could have a challenge in terms of neurological function or neurocognitive function. Physical causes. You can have an accident as a kid. Your mom could drop you on your head. I did my daughter. That's a horrible thing to admit, but you feel really bad at the moment. She's fine now in college. I think she'll forgive me if she ever sees this video. She'll probably give me heck for it. But There are cancer therapies, so that are acquired as a consequence of other things that you experience in life. And this is a big one. This is how I started my career. Kids that have cranial radiation never recover. In fact, they get worse and worse cognitively as time goes by if they survive. And that's in part due to a stem cell defect that's caused because they have a robust inflammatory response, so an immune-driven defect in their late developmental processes. Genetic disorders, we all know about these. A bunch of idiopathic diseases where there isn't a gene that's been identified. There may be risk factors. ADD, autism spectrum disorder, that's what we'll talk about today, but many others. They're immensely complicated. A lot of the phenotypes overlap in the individuals who suffer from this. And particularly in autism, there is this statistic that just scares the bejesus out of me. 2010, 1 in 68 children from the CDC studies are born and are diagnosed ultimately with ASD, autism spectrum disorders. And I think this crowd probably knows more about why those numbers are going up so much, but it seems to me as a scientist that it's really important to understand today what might be driving some of that really amazing 1 to 2% of all births. And I was just learning today that the new stat is if your first or one child has autism, there's a 25% chance the second will too, which is just an immense risk, an immense burden on both the family and healthcare in general. So I love this slide. It's a blog that I found online. It's a mom saying, this is what I am if I have autism. And so there are things like lack of eye contact, motor stereotypies, we call them, stereotypies, where there's a repetitive motion or unusual movements, very narrow interests, don't like change, very hard time interacting socially. And so there are a lot of features that you and I would immediately recognize because we're humans and we communicate that way. Scientifically, there are the 3 core components to autism. There are the social deficits, the language impairment, and repetitive behaviors. What are the causes? And I think you've probably heard many, many lectures at this institute in general and Davis at large about what could cause autism. What are the risk factors? What is the etiology? Well, there are many traits that run in families. So you say that these traits show strong heritability. That means that the genes are playing a role. You're inheriting a genetic predisposition for a particular trait. And that is very strongly true in autism. So most genetic risk is not single genes that have mutations, but a constellation of common variants. So these are differences in coding or basic DNA sequence that differ between individuals, but by themselves really have no impact in who you are or what your functional outcome is. And there are estimates about how much of you can account for autism in the genetics. 52% is heritable. 45% is other factors in this particular study. Familial risk in twins, 50% of monozygotic or genetically identical twins both have autism. The rest of the time they don't. So it's not just genes, it's genes plus. And so what are these other factors? And they're varied. And I think the most interesting for an old guy, I was old before I had my daughter, I was educated and I had better income. That is, in fact, one of the most recurrent demographics that are associated with an increase in incidence. Here at Davis, you're looking at agricultural pesticides and other environmental toxins have an effect. And of course, Judy and Irva have both been looking at that. There are other things that are less well defined, like just adverse events during pregnancy. Uterine bleeding is one. So if you have bleeding during your pregnancy, and in particular in the twin studies, bleeding is associated with very high discordance. So one twin gets it, the other one doesn't. Perinatal hypoxia, that's the other non-genetic factor that leads to discordance in twin studies. Infections, viral infections, and if you infect the amnion or the fetus or the membranes, the outcomes can be really severe, including attributes of autism. But then there are other types of immune events during first and second trimester, and this is what I'm very interested in, what are the characteristics of these immune-related risks, and is this cause? Or is it just coincidence that you can have an immune phenotype and it just so happens that you also have an increased risk phenotype, but the two don't crosstalk. So it's not the immune-driven changes in brain development that cause the effect. I think it's cause, and I'll show you a bit of data. So first, some of the work that in fact has come out of this group. Judy has been very productive in telling us exactly how dangerous it is to be pregnant and a normal human being. So things like asthma and allergies, I don't think the autoimmune variables have panned out. But this was an early example from the Kaiser cohorts that just immune events that are not necessarily infectious but involve mom's immune response might have a risk. Influenza and schizophrenia, 7-fold increase if there's a diagnosis in first trimester. That's massive increase. I couldn't believe that when I read that paper. There are other more recent studies now, 30% increase with any diagnosis of infection if you are in the hospital. And a lot of this is you're admitted to deliver your baby, and then a cryptic infection is then observed or seen. And so there's a lot of third trimester waiting to this, but it doesn't necessarily need to be third, first, or second trimester, any time during pregnancy. 50% risk of ASD, and again, Judy is giving us a fear of life in general. Cytokines are elevated. So, sorry, I don't mean to pick on you. So here's the challenge, right? I'm a scientist. I want to try to understand the molecular underpinnings of this association. Is it cause or is it consequence? And I think at least 2/3 of you in the room will agree with me that these are not equivalent model systems. Okay, humans are different than mice, and so as a scientist who's working on a mouse, you have to find those features of the mouse that might be informative to the way humans work. And so what are those? Mice socialize, but instead of making eye contact and using body language that we understand, they're more olfactory. They like to sniff their environment and they interact through primarily olfactory cues. Repetitive behaviors. You might have an activity that a kid will engage in over and over and over and over again. And for mom and dad, this is horrible. In a mouse, you can ask, do they bury marbles? And so there's a natural behavior. They will bury a few marbles, but then there are either genetic or environmental effects that can change their marble burying behavior. And so they bury more or fewer marbles. Learning disabilities. Kids, we have all experienced conditions where we have a hard time learning. Mice, maze paradigms of various sorts, social learning, contextual learning, fear learning, spatial learning and memory. Where did I leave my keys last night? I can't find them and I've got to get to work. That's a kind of learning and memory that we can measure in a mouse as well. Hyperactivity. You know, kids, you know it because you want to yell at them and tell them to stop. A mouse, it's a little bit more difficult. So this is just an open box. You can use a video recording system and you can tell that this is the normal exploration. There's a mouse that's slightly hyperactive. It's going all over the place. Here's a mouse that's less active. And so you can draw some parallels between these two. So our research paradigm takes advantage of some of those similarities. They're not human traits, but they're traits that mimic some of the features of autism or neurodevelopmental disorder. So a mouse mom, we give an immune stimulus rather than giving a full-blown infection. We actually use proteins or compounds that are isolated from viruses or from bacteria to trigger a non-infectious response. And so it would be identical to being infected, but you can very precisely control the amplitude of the mom's response, the duration, how many times you want to stimulate this. So we do an artificial immune stimulus, first or second trimester equivalent. We look at the outcomes, and because if mom is stressed during pregnancy, the mouse mom changes the mothering behavior after the babies are born. And we don't want to measure mom's behavioral differences. We want to precisely measure how the fetal brain and the offspring behaviors change. So we have to take them away from their birth mom, give them an adopted mom. It's called fostering. And those moms are completely naive. They have gotten pregnant, they've had their litter, and we just mix in the adopted babies with the normal babies, and then they get taken care of by a mom who hasn't been stressed. Then, of course, we test them. So this is the immune response. Lipopolysaccharide is just bacterial cell wall preparation. It binds the same receptor in immune cells that bacteria bind to and activate the innate immune response. And so happens that if you get a bag of E. coli contaminated spinach, that's the thing that makes you feel sick. And LPS causes similar sickness behavior in humans as it does in mice. And I think you recognize all of these things as the day after your bad potato salad experience on Sunday afternoon at the ballpark, right? So you can't move. You're not willing to take a shower in the morning because you feel too bad, fever, reduced food and water because you're never going to keep it down if you did. And you lose a little bit of weight, mostly water weight, and it lasts 24 hours, 48 hours. Really severe food poisoning or bacterial exposure would last maybe a few days. Exactly the same thing happens with the mice, and you can make them really sick to the point of death if you give them enough of the compound, or you can make it really mild so that the molecules that are signaling in the mom to say, whoop, we have an infection, we've got to fight it off, are just barely detectable. And so from endotoxic shock to a 3-day flu-like down to we can't really measure any difference in the behavior of the mom. And at the molecular level, it's really hard to begin documenting that an immune stimulus actually happened. You can, but it's pretty subtle. So in a mouse pregnancy, and for those of you who are curious, this is a C57 black 6 wild type mouse. So inbred, all the mice are genetically identical. There's nothing different from one mouse to the other except for the treatment that you give it. So no genes involved here necessarily because they're all identical. Mice have a pregnancy that's about 19 days, 20 days long. So the middle of the pregnancy is day 10. And before the middle of the pregnancy, if you give low dose to increasing dose, it's so severe, the pups are so vulnerable that everybody dies, except with the very lowest doses that you give. So from 30, about 75% of the pups are just lost overtly. So the pregnancy in the mouse can't tolerate even the most mild challenge that we give. As the pregnancy progresses, then the pups get more and more resilient until at about E13 and a half, sort of approaching the last trimester, then suddenly everything is pretty resistant. So mom and the fetuses and everything survive to term, and everything turns out relatively well. So earlier in the human studies, there was a thought that maybe first and second trimester were the critical periods where risk is most acute. And so we picked a time point that's kind of in the middle of mouse pregnancy. We picked a dose where about 25 to 30% of the pups actually didn't make it. So we know that it's a pretty significant impact on the fetus. But then you have enough fetuses that are developing all the way to term. They grow up to be young adult animals. And so this really is the mouse equivalent of a dose of bad potato salad. And you all know what that feels like. You're out of the running for at least a day. You're not great the next day. I probably shouldn't relate this story. I won't. But it has to do with bad chicken salad at a recent Stanford event, but and it was bad. Anyway, so that's the level that we're looking at in the mouse model. It's not too bad, but you definitely know that you're sick, and you'll get over it pretty quickly, but the pups go on. So what changes in the mouse? And this is a paradigm that we just measure sniffing. This is the social interaction, and Jackie Crawley, who is here now, developed this paradigm. It's called the three-chamber social interaction paradigm. And there are cups, wire pencil cups. There's a mouse here that the experimental mouse has never been introduced to. So this is the novel mouse. And we can measure how much time the mouse interacts here with another mouse, social interaction, or whether it likes to interact equally well with something that's inanimate, so just curiosity about another object. And you know, these are fairly rudimentary measures. You use video tracking. You can tell that the mouse is interested in that mouse. They'll also go and explore the other cup. And if all you do to the mom is give this mild transient illness, then the offspring socialize less. They just spend less time interacting with the mouse as opposed to the cup. They can remember that they've interacted with that mouse, and they prefer then to interact with a new mouse if you give them two choices. So there's nothing wrong with their social memory or the preference for social novelty, but they just don't interact as much. So they're a little bit less social. So repetitive or compulsive behavior, you can see from the figure on the right that the animals that come from an LPS challenge pregnancy are much more compulsive about burying all these marbles. And in fact, they get close to the 15 marbles that are in the cage within a 10-minute period. So that's really quite different. Now, this is a more complex learning and memory task. This is water maze. It's a horse watering trough with water in it. And under the surface, we have a little bit of children's tempera, non-toxic paint, just white paint in the water. So you can't actually see it, but under that dotted circle, there's a platform just under the surface of the water. And there's actually a mouse here, and mouse here and here. And this is 6 sessions during a day where that mouse is trying to learn where the platform is. And by the 6th session, the last session of the day, the mouse knows exactly where it is. You know, during the middle sessions, it kind of knows, whoop, almost found it, there it goes. But the first session, it has no idea where that platform is. And so you can measure the decrease in number of seconds it takes to find the platform over time. And if the animals are from an LPS pregnancy, they can learn, but they never get as proficient in a day of learning as the others. So they have a subtle learning deficit. So social impairment, a small one, compulsive repetitive behavior phenotype, and a learning and memory impairment or a spatial cognitive impairment. So those are the features that are different. Everything else we have measured is identical. So these are pretty normal animals, but they do have differences. The differences are evoked purely because they experienced this immune event during pregnancy. No genes, nothing else involved. Mom was mildly ill, and then the kids are different. So to cut a very long story short and point you to a couple of papers, the mechanisms that are involved involve the mom's immune response, how many cytokines are produced, what kinds, the amount that are produced. It also involves the placenta, and I'll show you a little bit of data on that. And then cytokines and other immune molecules actually get into the fetal brain, and there's very likely to be a direct effect of these immune signaling molecules on brain development. So it's fairly complicated, and let me go over some of the observations. This is a row of mouse placentas, and there are 6 to 10 fetuses in a mouse pregnancy. And we're looking at the uterine artery, the uterine vasculature, and the blackish rims that you see around each placental disc, that's frank hemorrhage. So LPS at a relatively low dose is causing a little bit of hemorrhage. At slightly higher doses, you're causing more bleeding in the placenta. If you go above a certain threshold, the pregnancy aborts. The placenta is essentially unrecoverable from that. But with a low enough amount, then the placenta heals. It might scar, and the pup goes on to term, but the placenta is never the same. During that particular time, you can use ultrasound just like you would use to monitor your baby during pregnancy. This is Doppler micro-ultrasound, and we're looking at the blood flow in the mouse umbilical artery. And each peak is the heartbeat of the fetal mouse, and it is quite rapid, and the area under that peak is equivalent to the amount of blood that's being pushed with every pump. In the immune model, you get a very severe decrease in heart rate, and the volume decreases. So you can imagine if you have a placental injury, you're not getting as efficient a transport across the placental membranes to the fetus. The fetal heart rate goes down, and the amount of blood circulating in the fetus is reduced. You could guess that the fetus might go transiently hypoxic. And this is a cross-section of the developing fetal brain. And these structures here are the early cortical structures, the outer structures of the brain. And we've used a chemical that will bind to tissues if the oxygen drops below 1%, which is already very low. So more red is more hypoxic, more low O2. And so in a normal developing animal with no placental injury, this is slight traces of low oxygen in the fetal brain, that's normal. But then when you have this immune challenge, there's a big increase or big decrease in perfusion and an accompanying increase in hypoxia. If you look at different brain regions, the cortex seems to be one of the most pronounced, where there's a big increase in the amount, the fraction of tissue that drops below 1% and becomes stained. But other areas also have signs of increased hypoxia. Normally, if you have a hypoxic event, like a stroke, ischemic stroke in an adult brain, a lot of cells die. That doesn't happen in the fetal brain. And this is something we're really curious about. But here, the red marker tunnel means a dead cell. And in a typically developing brain, each blue dot is the nucleus of a cell in the cortex. And you can see there's not any more red in the LPS model than there is in the typically developing brain. So it's not that hypoxia is killing cells, and that's leading to the changes in brain structure. It's something more interesting and more subtle than that. And one of the things that we can do is to, with our knowledge of how the brain is built during development, we can ask what components of normal developmental biology are altered. What can we measure that might change? And this is early work from Arnold Kriegstein's lab when Stephen Nochter was there. And so There are a population of cells called radial glia. They're the stem cells of the developing brain. They have a connection on the inner surface and on the outer surface, and they divide. They make a daughter cell that then differentiates. It's no longer a stem cell, and early in brain development, they migrate toward the outer surface of the brain to become a neuron. Stephen's work elegantly showed, has beautiful movies of these, that sometimes these divide more than once as they're migrating out. And these are intermediate progenitors, and they're amplifying themselves to make more neurons. And so that's how you start with a few stem cells and make the bazillion neurons that you have in an adult brain. So cell division, the decision to differentiate as a daughter cell and become a neuron, migrating out into the brain to develop a thicker and thicker brain, these can all change. And so there are very sophisticated ways of labeling the brain to mark cells that divided at a particular time in pregnancy. And so we've used one chemical called IDU. It integrates into DNA just like one of the nucleosides. And we injected at E12.5. And so those are all the white dots, the white nuclei that you see here. The next day, 24 hours later, we injected a second DNA analog. And those are all the red dots here. And I think you might perceive that some of the green dots are just green, some of the red dots are just red, but some of them are both green and red. That means, red and white. Sorry, I'm getting my colors mixed up. If it was white and then divided again, it would be red and white. If it was white but didn't divide again, it would only be white. The green marker here, Ki67, those are cells that we know can divide again, so they're not terminally differentiated, so they haven't completely exited the cell cycle. So if you're white, but not red and not green, you can't re-enter the cell cycle. It means you permanently exited. So we call those cells the quit fraction. If you're labeled and still green, it means you can potentially re-enter the cell cycle again. If you're double white and red, you did re-enter. So that's the re-entry fraction. So it's a complicated way of saying if you're a stem cell, and you want to make another stem cell, what you have to do is divide and maintain your stem cell phenotype. You have to divide symmetrically. You end up with two stem cells, or at least one of the daughter cells is a stem cell. If you decide to exit and no longer divide, you're no longer a stem cell. And so a decrease in re-entry and increase in quit means you're depleting the stem cell pool. And cells are differentiating too much. And one of the hallmarks of a symmetrical or an asymmetrical division is that these cells with a contact on both surfaces will undergo S phase. They'll take their cell body all the way down to the inner surface of the brain, and then they'll undergo mitosis. And most of the time, they divide symmetrically. So the two daughter cells are right next to each other. Occasionally, the cell will be a little bit tipped, and only one daughter cell inherits the attachment. The other daughter cell, as it divides, loses the attachment, migrates up to become a neuron. So vertical divisions or symmetrical, self-renewing divisions in large part. The tipped ones are asymmetrical or differentiating divisions. So we measured one way, quit fraction and re-entry fraction. And what we find in the LPS is that this anatomical arrangement, the cell polarity that defines symmetry or asymmetry of division, the cells tip more. They've lost their polar signaling that helps them to maintain stem cell phenotype. And so a couple of independent measures are telling us that we're disturbing the symmetry of stem cell division and fundamentally depleting a stem cell pool. That's one of the phenotypes that happens just from that mild immune response. So the other thing that we can measure is, well, if you're now making more of these differentiated, fully committed, the quit fraction cells, what do they become? They have to migrate up. If you're born on day 12, that means you need to generate a particular type of neuron in the brain because those are the layers that are being generated at that time. The other thing that has to happen is you have to stop migrating in the right place. And if you look at labeling in the fetal cortex and then later try to find out where those cells are in the adult cortex, you'll realize right away that if you're born on a given day, you make cells that occupy one layer. They're one place in the brain. If cells born on this day ended up down here, they would be in the wrong place and perhaps doing the wrong thing in terms of the circuit and function. So in the fetal brain, we have markers that recognize the outer surface, and that's where the cells should stop migrating. And this particular molecule is called relin. It is one of the stop signals. And the stop migrating signal is actually reduced in the LPS brains. So there's less of a stop signal. Certain types of neurons are less abundant, so you're just making fewer of them. So this is 24 hours after the immune challenge, and this is several days following that. And so this particular neuron, some of the early born cells will stop and they inhabit the deeper layers. Another set inhabit the middle or the upper layers. In the LPS model, you're depleting the lower layers and you're overrepresenting the upper layers because there isn't a stop signal. So they're ending up piling up on the top surface of the brain and creating a laminar disturbance in the way the brain is built. You can confirm this in the adult animals. So in this particular LPS model, this subtype of neuron, if the bar goes left, means there aren't enough of them compared to a normal animal. And each bar is 1/10 of the way up the cortex. And so you can see throughout the entire cortical layer, all layers are depleted of this particular neuron type. This type, bars to the right, has now made too many in the upper layers. So they're overrepresented. This is another type of neuron. Both upper and lower are slightly underrepresented, and the middle is kind of fine. And then neurons that are born much, much later are not affected at all. Very, very small impact. So there are some very substantial changes in the way the brain is built, disturbances in stem cell biology, disturbance in the migration and fate of the daughter cells that are being produced. We don't know what happens to the circuitry, and I have a postdoc that's working on that right now, but you'd expect that if cells are too few or too many or in the wrong layers, the circuits aren't going to work the same way they should in a normal animal. So I won't talk to you about that, but I will do this. Every mom, most moms, might have a mild illness during pregnancy, but very few, well, let's say 98% of us don't have autism, right? More than 98% of moms have an illness. So what's going on? And what we're interested in asking is whether any of the genes that are not alone sufficient to cause autism but are known risk factors, whether they might interact in some way to make the pregnancy more vulnerable. And you can imagine the gene could operate in the mom, right? It could operate in the placenta, could operate in the developing brain, or all three. And so that's essentially where we started. Organisms don't waste energy. So if you're an evolutionary biologist, you realize that one gene plays many roles, and it depends on what time the gene acts and in what tissue the gene acts. And so you don't just have one brain gene. You have a brain gene that causes a change in brain development during development, but the same gene might have a role in the immune system or maybe the placenta. And so this was our speculation that some autism genes that regulate function in immune cells, placenta, and brain all at the same time, if they synergize in the immune model to make each of those biological compartments have a more risky response to the immune challenge, then you could have synergy and the outcome could be really worse. Turns out that if you look at the placenta and ask, well, is there a set of genes that might be related to placental bleeding or vulnerability? This you've already seen. Pups are lost with very low doses in early pregnancy. Right between E12 and E14, they kind of undergo a transition where everything is really resilient. And in fact, these placentas bleed, these don't. This is the gene expression pattern. And for those of you who are used to looking at that, blue are genes that are low expression, red are genes that are high expression. And each tiny row is the expression, the amount of RNA, gene expression, that occurs at 8, 9, 10, 12, 13 days of gestation. I think your eye can tell that of the genes that are regulated twofold or more, there's a set that start low and suddenly switch to high. Another set that start high and suddenly switch to low. And surprise, surprise, happens to be right across that boundary in time when the placenta is either really vulnerable or very resistant. So among these genes, are there any in the autism gene database that we could ask, well, could it potentially be involved in placental vulnerability? So placental genes are brain genes, and is there a subset that we should be looking at? So Nora Vivanco, an immunology grad in the lab, said to herself, well, there are a subset of genes that are really well validated. So they've been identified multiple times in studies. There might be some animal evidence that they change brain development. And she picked a set that are autism related or schizophrenia related and said, how many of these express in the immune system, in the placenta and the fetal brain? 6 autism genes, 2 schizophrenia genes. Of these, there were 4 that have mouse knockouts. And so what we did is asked whether a loss of the gene influenced the survival of the pups. 4 out of 4 autism genes that meet the criterion had 0 surviving fetuses, totally wiped out. So big increase in vulnerability. 1 out of the 2 schizophrenia genes had the same phenotype, and the other one is in fact maybe even more resilient for whatever that mutation is. So I'm going to tell you about just one of those genes. It is a neurotransmitter receptor gene, and the gene itself is called GABRB3. It's the beta-3 subunit of the GABA-A inhibitory receptor. In the brain, it's the primary receptor that mediates the quenching of activity in neural circuits. So inhibitory interneurons produce GABA, the ligand for this receptor. GABA then prevents other cells that it's acting on from firing. There are excitatory neurons. They make another neurotransmitter, glutamate, and they can turn up the rheostat on activity. But this is the primary turn down activity mechanism in the brain. It's part of a cluster of genes that is included in a copy number variant, and this is a very interesting region of chromosome 15. So if you have a deletion across the domain, you have Angelman syndrome. A maternal loss is Angelman, paternal loss is Prader-Willi. A duplication or triplication is autism risk. And one of these genes we're going to focus on now, none of the others, but it tells you that some of these and the related family members might be interesting in a variety of neurodevelopmental disorders. So it has some interesting maternal and paternal imprinting phenotypes that account for the differences in phenotype in the offspring, but it also is a receptor that's expressed by immune cells. And if you lose receptor function, the amplitude or the aggressiveness of the immune response is higher. If you have receptor function, it tunes it down. So the same autism mutation that causes a loss of function could also cause an increase in immune signaling or immune vulnerability. So that's the question that we asked. And the reason why we picked it out of the autism genes is that the subunit gene actually is just one gene for 5 subunits of the whole receptor, so there are other genes involved. And among those genes are some accessory proteins that if they're missing, the receptor doesn't work. If they're present, the receptor assembles and functions very efficiently. This is placental gene expression of the subunits and the accessory proteins. Early in pregnancy, the assembly, the complex is low. Placenta is vulnerable. Late in pregnancy, the complexes, all the components are present and abundant. And so the theory here is that the function of the receptor may be related to placental vulnerability, as well as may function in the immune cells to modulate the maternal immune response. So one of the first things that you can do, because this is a receptor that's been studied for decades or maybe even a century in neuroscience, there are drugs that are agonists or antagonists for the receptor. And you can take some immune cells out of the peritoneal cavity of the mouse. These are macrophages. You can treat them with LPS and measure the cytokines that are elaborated into the culture media. So they secrete the cytokines. Same thing happens in the mom. They circulate and activate downstream immune events. If you put the pharmacological inhibitor of this receptor, many of those cytokines increase their abundance. So the cells are directly responding to an inhibitor by increasing the amplitude. If you activate the receptor, then they decrease the expression of the cytokines. So that's proof positive that the receptor is working, that you can pharmacologically modulate it. What happens if you just take a wild-type mom and ask, well, if you inhibit the receptor, what happens to placental pathology, that bleeding and necrosis? And this is a wild-type mom. This is a wild-type mom with the inhibitor. So the amount of tissue damage goes up many-fold if you just pharmacologically manipulate that particular receptor. At higher doses, when you have high damage, you can actually protect with a drug, actually clinically relevant drugs. And so this gets to be more and more interesting and starts to add some weight to our model. Here's wild type versus LPS plus umbilical artery blood flow. Again, this microdoppler ultrasound. Here's what happens to heart rate if you have the agonist, the upregulator of this neurotransmitter receptor. The heart rate doesn't go down. So the pup and the perfusion is protected just with a drug, and you're modulating these compounds. So in the mom, let's say you knock out just one copy. The mom has the other normal copy, so it's a model of partial loss of function you might find in a maternally inherited gene. So by 2A ANOVA, the effective genotype is really pronounced. So mom elevates cytokines, some more than others. So yes, this gene mutation is affecting the maternal immune response. It's elevated. Okay. What about the amount of necrosis in the placenta based on the wild type or the het, loss of one allele? And so I'll go really quickly through a series of measures here and show you what the effect of the mutation all by itself. So this is in a typically developing pregnancy. Nothing else happens. This is just the mutation. Not really any difference. This is what LPS does. It increases the damage in the placenta. This is what the gene plus LPS does to the placenta. So there's an additive effect here. Then you can ask, does the baby have to be mutant? Or can the mom all by herself be the cause of this? And so here is a set of experiments where the mom is wild type and the pup is wild type. The mom's mutant, but the pup is wild type. The mom is wild type, but the pup inherited the gene from the dad. So this is the effect on placental injury if it's just mom. Cytokines are higher a little bit, maybe a slight increase, but it doesn't reach significance. So mom's not sufficient, okay? What about if dad's the donor? This gene now here is acting in the placenta to increase the vulnerability of the placenta to a normal mom's immune response. So there's a lot going on, and I have to cut the story short, but we're looking at how immune cells are trafficking in the placenta, and whether or not there's some interesting immune mechanisms that are going on there. But what does this set of increased vulnerability features do to adult function? So here's the social interaction paradigm. That's the effect of the mutation. This is the effect of LPS, the white bars. That's the effect of LPS plus the mutation. So you can imagine 50% of the time these animals are not interacting, well, they're equally interacting with the empty cup and the mouse-containing cup. So essentially, these animals don't show any social preference. Marble burying, here's the mutation all by itself, LPS all by itself, and the combo. So you can see that there's an increase. This is a more complex learning and memory task. And essentially, all of the conditions except for the combo aren't affecting this particular. You have to have the combination of the immune challenge plus the mutation. So I have to cut it short here because I don't want to bore you too much tonight. But 50% of autism is known genetics. Maybe some more as we begin to explore parts of the genome that we didn't think were important, the dark matter of the genome. But 50% right now is other things, and those other things include adverse events during pregnancy. So we have immune events, and we can check that off. The mouse model has that. We have uterine bleeding. The mouse model recapitulates that as an accompanying feature. There's hypoxia. We can recapitulate that. And then along with all of those phenomenon, we get some specific changes in both the molecular control of stem cells and cell fate in the developing brain, loss of polarity and mitotic spindle alignment, reduced relin and overmigration, and depletion of certain subtypes and overrepresentation of others. So I leave this to you. Is it cause or consequence? If you're a mouse, I know how I would answer that question. If you're a human, I'm waiting for Judy to answer that question for me. So in summary, illness all by itself does change brain development. And in the mouse, you can do it over and over and over again. It's reproducible. Kind of flippantly, what your mom ate can change your mind seriously. And at many levels. It's not just in this immune model. I think this is an important underexplored area of biology, maternal health, gestational health, and what that means to the outcome of the child, or perhaps the grandchild, which is another interesting area of study. So strong synergy in this one gene that meets this really interesting set of criterion: immune, placental, and brain function. And the behavioral consequences, curiously, are more pronounced in the male offspring. The placentas have more damage if you're a male fetus. So the 4-to-1 prevalence of autism in males is an interesting correlate there. Gene by immune interactions, I think what you can take home is that the autism brain genes do other things. They're also immune genes and placental genes. And whatever other biology you would like to study, there's very likely a role for that mutation in more than just the way the brain develops intrinsic to the developing neurons and glia. So maternal illness alone, I think it's just a source of diversity when we look at the human population. But if you put it together with a risky mutation, then you get a context that's really quite severe, dangerous, in fact. So with with that, I'll close. I would like to introduce my group. These are the people that come up with the ideas and do all the hard work. In particular, two prior postdocs, Pam Carpenter and Ursula Hadich, and current fellows, Amy Moon and Harion Lee, and more recently, Brooke Babineau, who was here and is now helping us with many of the behavioral paradigms. I was so happy to steal her away from where she was and get her to come to my lab. But it's a great group, and welcome inquiries from any interested parties, and I'm happy to answer questions. Thanks.
Do you know which cell types in the placenta are expressing the GAB receptor beta 3?
Yes, all at some level. And so, yeah, it is surprising. And you don't, oh, pretty neuron specific, yes. We're looking by transcriptomics and by immunofluorescence, and you can see signal for both. So the beta 3 subunit and the subunits proper are quite low. The accessory proteins required for proper assembly and maintenance of the active pore at the surface are much more abundant. But the placenta expresses much lower levels than the brain expresses, and immune cells are low but kind of intermediate relative to those two.
Do you think the accessory proteins are actually doing the action on very low levels of the...
So I think it doesn't take much receptor to change so it's a chloride channel, it changes calcium content in cells, and it doesn't take many receptors to do that. What is important is whether they work or don't work. In other words, if you don't have the accessory proteins, you can have all of the receptor subunits present, but the receptors won't work efficiently.
Given your research and the variability in responses to viruses, I was wondering what your thoughts were about their giving immunizations to women during their first and second trimesters of gestation. Because a lot of the mothers that I've interviewed who have children with autism were given vaccines at that time.
I would have a very subjective and non-scientific answer to that, but somebody like Judy would probably have a much more specific answer to the association, if any. So yes, when you get a vaccine, it comes with an adjuvant and it causes a local immune response. It's much, much smaller and very localized to, for example, the skin if you get a dermal injection. We're talking about a more widespread immune response or cytokine distribution. So we're injecting the compound directly into the peritoneal cavity of the mom. And for a small amount of compound, we get a pretty big response. So my impression, my guess, is that the amplitude is so small with the vaccine that it's not relevant at all for this type of model, that you need to feel really ill if you're pregnant and you have a really high risk genotype, that it takes a pretty significant event that you're going to notice it, and the vaccine is not that.
I was just curious more about the placental hemorrhaging that you saw, because I know people who have worked on the MIA model have found that some offspring might exhibit greater behaviors or greater immune responses than others. And I was curious, when you were just visually inspecting the uterine horn, if you noticed that there were some placenta and babies that were more affected, and if so, was that region consistent? Was there a particular spot in the uterine horn that was always more severely affected than others?
Yeah, that's a great question. So in mice, you have two uterine horns, and the placentas and the fetuses line up along the horns, half on one side and half on the other side. And there is evidence that where the fetus is in the position within the uterine horn makes a difference. And in our studies, anecdotally, what we find is that the position in the horn isn't as important as position next to a very severely affected placenta or fetus. And so they tend to be next to each other. And a fetus or a placenta in the exact same pregnancy that's far away could be completely normal. So in a litter of 10 animals, 60% of them can have signs, visual signs of hemorrhage in the placenta, and the other 4 might have nothing that you can see. So it does vary, but the place, the actual position in the uterine horn, we haven't scored enough to really say whether that makes a difference, but it's not obvious.
Do they always inject on the same side, or do your postdocs?
I do, but I don't know if my postdocs do. Yeah, I'll have to ask them. My guess is that we try for midline, so that's the protocol that we use, but it doesn't mean that the uterine horns might be off center and maybe one would be closer to the injection than the other, and that's definitely a possibility.
Very interesting research. Just a little speculation. These risk genes that in the presence of the heightened immune response, do you think it's possible that they might have beneficial effects in the absence of the immune response? This idea is kind of coming from some of the resiliency research.
Yeah. So I think the question is whether or not a mutation that might increase gestational risk to an immune event might actually be beneficial to our species because it makes us more aggressively fight off pathogens or have resilience to infections that would otherwise be a problem. And I think that the answer is obviously that some genes will have that as an attribute. Whether or not, so let me answer it this way. The work from the 2 immunology graduate students in the lab, Nora Vivanco and Amber Moore, are showing that the immune cell trafficking and immune status between mom and the placenta is really unique during pregnancy. So mom's immune status changes. That's induced in part by the fetal placenta. And that interface is really an interesting immunological barrier where it actively prevents mom from recognizing an allogenic fetus. So when you stimulate with the immune challenge, everything changes. There are massive changes in the placenta in the balance of tolerizing or effector cells and things, and how these genes act in subtly disturbing that balance is a hypothesis that maybe they do, and you're closer to a tip-over point where tolerance is no longer maintained, for example, as a mechanism. We don't know yet, but that's speculative and supported in part by the really interesting changes in how the immune cell components traffic and change in the model.
Did you see also the same prevalence in your model with the male pups, or the location that they were located in the uterine horn? Because at least in rodents, even being the alpha is determined by the position in the horn, because the hormonal milieu changes. Did you see also something similar in your model?
Yeah, I think if I understood you correctly, that's similar to the question that was just asked about. Naturally, there are differences in fetal support depending on where the fetus is in the uterine horn. So hormones change depending on whether you're proximal or distal in the horn. And we don't see a really robust positional effect. Okay. So I can't tell you, so if we're just looking by eye, it's not always the distal end or the proximal end of the uterine horn where we see the injury. It's throughout. But what we often see are clusters of severely affected animals.
Thank you for a great talk. My question is going back to your earlier slides related to the increase that we're seeing in autism. So if the mechanisms that you're talking about may play a role in that, then does it mean that mothers are getting infected more often, or does it mean that their immune systems are reacting differently than in the past, or?
So I've had this conversation with many individuals, and I don't know. So I don't think that we're exposed to more things. What I do know is that in this time period, the use of broad-spectrum antibiotics has gone way up, and it may be that people are, this is very speculative, and I think the use of antibiotics is such a critical part of care. But essentially, if you have any kind of an immune complication in pregnancy, one of the first things you do is you treat. And it might be that as a species, we have been exposed less, we have less resilience overall, and just because of the way we treat exposures. That's so speculative that there's basically no data that I could point to show that, but that's a possibility. The fact that we try to quench the exposures, and we simply, as an individual, may have had less exposure and have a higher response rate or less resilience to a low-level infection. But the increase can't possibly be due to exclusively due to a change in the immune state of moms or vulnerability of placenta in kids. Yeah, and so I couldn't even begin to estimate that fraction, and then whether that fraction has changed. One there and another one over here.
So thank you for teasing this apart so magnificently, and it's still hard to comprehend. But I was curious to maybe have your thoughts about the association with viral infections. And so CMV and rubella are, likely candidates because they go right there where the fetus is.
Yep.
I was surprised to hear about influenza, and could you maybe shed light on that type of viral infection, what the difference would be and how the common pathway might exist for effect?
So the question, if I heard you correctly, is there are several infections, CMV, rubella, that actually infect the fetus. And you could understand why an infection in either the fetal membranes, the placenta, or directly in the fetus, you can understand why that could generate a pathology in fetal development. But flu doesn't. So flu is systemic, it's in the mom. So how could that cause or increase risk of autism? And I think the answer is in the animal models. So we used a bacterial infection model, but you can do the exact same thing with the influenza infection model. Rather than using the flu, you use the genome of the virus or mimic of that, and that activates a virus-specific and influenza-specific immune response in the pregnant animal. That also is sufficient to cause changes in brain development and behavior. So we think that the mom's immune response is affecting the function of the placenta in general. We don't know necessarily what the specific changes are in the placenta. And we also know that some of the immune signaling molecules, the cytokines and chemokines, can cross the placenta, and they're elevated in the fetal brain where they can act directly in the developing fetal brain.
So if I understand I understand that stress can increase pro-inflammatory markers and cytokines, and I wonder if you've looked into that or thought about that or can talk about that and its relationship to autism.
I think about that a lot. The postdocs who are having families in my lab are thinking about that a lot, especially just before grant deadline or when we have a paper that we're trying to get back in. And my lab hasn't looked at that directly, but other groups have asked simple question: if you have mild stress during pregnancy, and the stressors are things like just tipping the cage a little bit for the pregnant mom, And the pups, when measured in these measurements, actually do show changes in behavior. What we don't really know is whether that's a developmental or a mothering thing, if the mom was stressed and has changed her mothering instincts, and that's what changes the behavior of the pup. So those variables haven't been teased apart. But it's really clear in the experimental models that what they call the ultra-mild stress paradigm can change the outcome in the pups.
This goes to your slide on neuronal migration. It was a little dense, a little bit too dense for me to remember every detail, but it appeared to me that deep layer neurons were more affected with respect to the mispositioning than upper layer neurons. And I wonder, first, is this the case? And second, is this the case because it's something to do with the LPS administration during the birth of the deep layer neurons, or is it just a universal effect?
So the data is in Carpenter et al., 2013. Go find that. But the question is, in the adult, in that slide that had so many bars in it, it looked as if, and this is a very astute observer, that maybe the deeper layer neurons were more affected and the upper layer neurons may be less affected. And that's absolutely true. And the reason I think that that happens is that the challenge is given during deep layer neurogenesis. And the immune response and the placental injury has healed by the time the superficial layers are being generated. And so they're born and migrate and populate the layers accurately, more accurately than the deep layers that had been born earlier.
And you have not made attempts to change the LPS administration protocol to see if
later
upper layer
or
neurons
earlier?
would be equally vulnerable to
Yeah,
this positioning?
we have not. And others have. So Urs Meyer has done LPS injections at different times. He hasn't done the anatomical studies to know if the disturbance is the same, but the behavioral outcomes are different, implying that the circuitry that you're affecting is a different subsets of circuits depending on when the challenge occurred. So an early gestational challenge doesn't give you the same features as a late gestational challenge. And it makes biological sense because certain structures are formed early, other structures are formed late, and it appears that the structures most affected are the ones that are in active development at the time of the immune challenge.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.